Hi friends, I am happy to meet you through this video. In this video, I am going to discuss about the classification of fungi proposed by Alexopoulos and Mims in the year of 1979. Fungi are a diverse group of organisms. They play an important role in the environment as decomposer, symbiont, pathogens, and sources of useful substances. However, the classification of fungi has been a challenging task for mycologists who have proposed various systems based on morphological, physiological, biochemical, genetical, as well as the molecular data. But one of the most influential classification of fungi was proposed by Constantine John Alexopoulos and Mims and in the Charles W. Mims in the year of 1979. They published their system in a textbook titled Introductory Mycology. In their system of classification, they based on the ultrastructure of the fungal cell wall, the presence or absence of flagella, types of sexual spores, mode of spore dispersal. Alexopoulos and Mims placed all fungi, include the slime modes, in the kingdom Mycetes of the superkingdom Eukaryota. Alexopoulos and Mims in their classifications, they divided the entire fungal kingdom into three divisions, eight subdivisions, 11 classes, one form class, two subclasses, and three form subclasses. They see this is the classification proposed by Alexopoulos and Mims. The group Mycete is coming, the fungi is coming under the kingdom Mycete. They have following characters. They are achlorophyllous. That means chlorophyll is totally absent. So they can't able to prepare their own food material. So the nutrition or the top mode of nutrition is heterotrophics. They depend other organisms for their survival. They are acellular, unicellular, or mycelial thalloid. Cell wall is made up of chitin. Food reserve is glycogen and oil droplets. Here, the division Gymnomycotia is subdivided into two subdivisions. One is Acrasiogymnomycotina and Plasmodiogymnomycotina. Gymnomycotina have following characters. They are plasmodial slimy organisms. They lack cell wall. They are phagotrophic nutritions. Phagotrophic nutrition is nothing but the intake of food by phagocytotic method. That means the process phagocytosis is engulfing food in the cell leading to digestion. They are dictyostelium. The division Gymnomycota carry two subdivisions. One is Acrasiogymnomycotina. The Acrasiogymnomycotina carry only one class. They are called as Acrasiomycetes. Acrasiomycetes, they are popularly known as cellular amoeboid slime modes, most abundantly found in upper layers of humus in well-established deciduous forest as well as in soils. Flagellated cells are absent. Mexamoeba aggregates to form a pseudoplasmodium. See, this is the life cycle of acrasids. The cells, they won't have any flagella. Second subdivision is Plasmodiogymnomycotina. These Plasmodiogymnomycotina carry two classes, Protosteliomycetes as well as Myxomycetes. The class Protosteliomycetes, they are primitive slim modes, mostly found in soil, dungs, humus, deadwood, tree bark, and plant remains. Myxamoeba do not aggregate prior to fruiting. The class Myxomycetes are true slime modes distributed worldwide, found on moist soil, decaying wood, and dung. Sometimes fruiting bodies are conspicuously colored. The second division, Mastigomycota. Mastigomycotina includes the fungi with absorptive nutrition, unicellular or filamentous mycelium. They are cenocytic. Cenocytic means the cell with multinucleate condition. Asexual reproduction, typically by zoospore formation. The division Mastigomycota carry two subdivisions, Haplomastigomycotina and Diplomastigomycotina. The subdivision Haplomastigomycotina carry three classes. They are Chytridiomycetes, Hyphochytridomycetes, and Plasmodiophroromycetes. The class Chytridomycetes, they are posteriorly uniflagellate zoospore. Hyphochytridomycetes, the flagella are present in the anterior portion of the cell. They are aquatic fungus. Plasmodiophromycetes, here they are parasitic with non-cellular multinucleate thallus. The second subdivision is Diplomastigomycota. Diplomastigomycota, they are the zoospore have biflagellate. They are meiosis gametangial conditions. Carry only one class, Oomycetes, found in a variety of habitats. Majority are aquatic, live parasitically on algae, water mold, aquatic insect, and other animals and plants. Mycelium is well branched, filamentous, and cenocytic. Cell wall contains cellulose. Zoospores are biflagellate, having one whiplash and another one is tinsel type of flagellum. Many members produce non-motile conidia. Flagellated gametes are absent. Fertilization caused by fertilization tube. Here you can see the whiplash as well as the tinsel type of flagellum. The third division is Amastigomycota. The Amastigomycota, the centrioles are totally absent. Spindle pole bodies functioning during cell division. Flagellated cells not produced during their life cycle. The division Amastigomycota subdivided into four subdivisions. They are Zygomycetes, Ascomycotina, Basidiomycotina, and Deuteromycotina. The Zygomycotina subdivided into two classes. One is Zygomycetes, another one is Trichomycetes. The class Zygomycetes are mostly terrestrial in habitat, living in soil or on decaying plant or animal materials. Some are parasitics of plants, insects, and small insects, while other forms are symbiont relationship with plants. Mycelium are cenocytic, forming septa only where gametes are formed. Perfect stage conditions is zygospore. The second class is Trichomycetes. These fungi are parasitic, found within the digestive tract of living arthropods and guts of earthworms. The host of Trichomycetes include marine, freshwater, and terrestrial arthropods. Asexual reproduction takes place by trichospores, sporangiospores, arthrospores, or amoeboid cells. Sexual reproduction takes place by biconial zygospore. The second subdivision is Ascomycotina, carry only one class, Ascomycetes. Here, they are otherwise called as cup fungi or sac fungi. Mycelium are septate. Fruiting body are called as ascocarp. Based on the morphology of the fruiting body, they are called as cleistothecium. That means they are closed. Perithecium, they have a thin opening. Apothecium, they are cup-shaped. Ascus act as a zygote. They undergo meiosis and form 4 to 8 ascospores. That conditions we can observe in the case of red mold, blue mold, as well as the aspergillus. The ascomycetes, carry 5 subclasses: Hemiascomycetidae, Plectomycetidae, Hymenascomycetidae, Labolbeniomycetidae, and Lochiloascomycetidae. Hemiascomycetidae, they are unicellular, filamentous, the ascomata are absent. Plectomycetidae, they are filamentous, ascoma clistocarpus. Hymenascomycetidae, they are also filamentous, as they are unitunicate. Labolbenimomycetidae, they are Hostorium or Rhizomycelium, obligatory parasitic on arthropods or marine red algae. Leucoascomycetidae, here the filamentous ascae bitunicate formed in ascostromata as a unitunicate perithecoid as well as the ascomata. The third subdivisions of division Ascomycota is Basidiomycotina. The subdivision Basidiomycotina carry only one class, Basidiomycetes. The Basidiomycetes are club fungi, mycelium septate, fruiting body are called as basidiocarp. Basidium act as zygote, undergo meiosis and form haploid basidiospores. Example, Puccinia. Here you can see the Puccinia spore, uridospore of Puccinia. And then the class Basidiomycetes carry 3 subclasses: Holobasidiomycetidae, Phragmobasidiomycetidae, and Teleomycetidae. Holobasidiomycetidae, here the mycelium with long dichoritic face. The Phragmobasidiomycetidae, they are longitudinally or transversely separate. Teleomycetidae, basidiocarp is absent. The last subdivision of division Ascomycota is Deuteromycotina. Deuteromycotina are an artificial group of fungi of which there exist about approximately 15,000 species, often referred to as fungi imperfecti because only their asexual and vegetative phases are known. They are also known as Deuteromycota, Deuteromycotina, the fungi imperfect, and as well as the mitosporic fungi. Mitosporic fungi is nothing but these fungi are unable to produce sexual spores and are therefore placed in their own separate phylum. Here the subdivision Deuteromycotina carry 3 form class, subclass: Blastomycetidae, Coleomycetidae, and Hyphomycetidae. Blastomycetidae consisting of yeast cells with or without pseudomycelium. Coleomycetidae, they have a true mycelium, conidia produced in pycnia or acervirus. Hyphomycetidae, they are true mycelium, conidia produced only on the gonidiophores. Thank you. Thank you very much for your